Good evening, everybody, and this is Hearts, the exclusive channel for NEET 2021 batch, introducing yet another super cool and super awesome class and lecture on the topic Plant Growth and Development, Class 11, NCERT, Botany chapter, based on the plan Physiology. So this is a very interesting chapter, how you can be able to get connected with the plants. Okay, generally for your NEET exam, only one question, only one question of 4 marks will be asked from this chapter, or it can go up to a maximum of 2 questions. So you will get a marks of 8 marks from this particular chapter, growth and development. That is, growth and development is a chapter. Plant growth and development is a chapter that constitute only 2% of the entire NEET paper. For NEET 2021, it constitutes only 21% of the entire NEET paper. Okay, so let's get started. So let's begin. Okay, all plant cells, okay, we are discussing about plant cells, right? So all plant cells are the descendants of zygote or fertilized egg. Zygote is a single cell, right? Condition is, sorry, diploid. Okay, it is a diploid cell, right? Zygote, fertilized egg, single cell, but it has 2 N chromosome, or it is a diploid cell. The zygote is the portion from which the plant gets mature. That is, right from the single cell, from zygote, the plant makes a journey. Right from the single, the plant makes a journey up to the plants. Is it clear? Okay, the plant makes a journey from the single-celled embryo to a mature plant. Okay, the zygote develops into a mature plant. Okay, the zygote or the single-celled organism just develops into a mature plant through different processes. For example, growth, differentiation, and they form certain parts of a plant such as root, leaf, branches, flowers, fruits, and seeds. And finally, when their lifespan is over, they eventually dies. And you have to make sure that the plants grows, the growth of plant is unlimited. That is, they grow continuously throughout their life due to the presence of certain tissues called meristem. Okay, 3 type of meristem we have studied in the chapter anatomy. That is apical meristem, intercalary meristem, and lateral meristem. So we'll discuss about all 3 type of meristem again in this particular chapter. Look how the zygote or the single cell gets first divides into a 2-celled condition, and this 2-celled condition changes into 8-celled condition, changes eventually into globular embryo, repeated times, frequently or repeatedly asked question for NEET. They will be giving this particular diagram and you will be asked to identify the structures labeled A, B. Okay, so this structure, this globular structure that I am marking indicates what? The embryo. And the structure that I am indicating with a green pen is what? It is the suspensor, which helps it to attach, okay. Finally, gains into heart embryo, heart shape. It changes into heart shape. Look, heart shape. And last, it turns into a torpedo embryo. They will be asking this particular thing. What are these? They are the cotyledon. So the part of the seed, suppose you consider this is to be a seed, and if you take into consideration the pea or the bean seed, you will be having a small white scar over here, and you can call that white scar as to be hilum. So from this, a small portion just grows downwards, right? So the portion that grows downwards grows as a root. So this particular portion is known as what? A radicle. So The radical in the seed just elongates to form root, whereas the other part that is formed above the soil or that forms like this. This is known as the plumule. This is known as the plumule, which indicates the shoot system. Okay, so the part of the plant above the soil is known as to be shoot, and the part of the plant below the soil, it is known as to be what? Root. Okay, is it clear? So there are certain type of tissues that is present at the tip of the root and at the tip of the shoot, which elongates the life-ending growth of the plants. So those tissues which are present at the top, that is on the tip of the shoot, is called the shoot apical meristem. And those cells or tissues present at the tip of root is called the root apical meristem. Is it clear? So let's move with the next one. Are you clear with the plant cell? The descendants of the zygote of fertilized egg. It gets developed into a mature plant through growth and differentiation. Okay, and eventually they get died. So next one, we are going to discuss in detail about our lecture number 1, that is the plant growth. So let's move on. So what is growth? Growth can be defined, okay, growth can be defined as an irreversible permanent increase. So it can be defined as an irreversible but a permanent increase in the size of organ. So increase in the size of an organ or a part of an individual cell. It involves metabolic processes that consume energy. Is it clear? So growth in plants is put forth continuously throughout their lifespan with the help of certain structures called meristem. Okay, meristem. So the type of meristem which helps in the elongation. Okay, the type of meristem which helps in the elongation of the plant. And that type of meristem you call it as what? Primary meristem. Primary meristem, and elongation in length or elongation or lengthening of the plant can be referred to as a primary growth. Can be referred to as a primary growth. Okay. Whereas increase in the girth of the plant. Increase of the girth of the plant is brought about by lateral meristem. Brought about by the lateral meristem. Okay, and it is called a secondary growth of the plant. So you have to study that the growth in plants is unlimited, whereas the growth in animal is limited. For example, all the changes that is associated with, one second, all the changes that are associated with growth is referred to as what? Development. So development can be mainly classified into 2 types. Okay, development. I think my pen is not working. Okay, development can mainly be classified into 2 types. Change the color of the pen. So the first type is morphogenesis. Second one is differentiation. Okay, first one is morphogenesis, second one is differentiation. What do you mean by morphogenesis? Morphogenesis can be referred to as the development that takes place in the shape of the cell, that takes place in the shape of cells or organ. Okay, the pen is drawing somewhere and I am writing somewhere. Okay, so what is differentiation? All the qualitative differences between cells. or may it to be tissues, or may it to be organs. So the individual differences between the different type of cells is referred to as what? Is referred to as development. Okay, so let's move with the next one. So this growth of plants is mainly due to the presence of specialized cells called meristem. Okay, the plant grows continuously throughout their life due to the presence of meristem. So meristematic cells, there are two types of cells, meristematic cells and permanent cell. Meristematic cells, they have the ability to divide and self-perpetuate. Whereas permanent cells are those meristematic cells, after a fixed amount of division, they get converted into permanent tissue or permanent cell that have lost the ability of self-replication. Okay? Capacity to divide and self-perpetuate. The growth when new plants, okay, this type of growth when new plants are added into the plant by the activity of the meristem. By the activity of the meristem is known as open form of growth. It is referred to as what? Open form of growth. That is a growth that takes place by the continuous activity of the meristem. Is known as what? Open form of growth. If you take into consideration, which color should I use? Yes. The plant at the tip of the shoot, I'm indicating it with a brown color. At the tip of the shoot, you can see the presence of what? The shoot apical meristem. The shoot apical meristem that continuously helps in the what? Lengthening of the plant. Helps in the lengthening of the plant. And if you take in case of the root, in the root also you can find what? The root apical meristem. And in the shoot, you will be finding the shoot apical meristem. Clear? So this is the shoot apical meristem and this is a root apical meristem. So let's get moved. Okay, plant growth is indeterminate. Plant growth is indeterminate. So there are two kinds of growth in the plants. The first one is the primary growth and the second one is a secondary growth, which can be observed only in gymnosperms and dicot. Which can only be observed in gymnosperms and dicots. Primary growth means by the activity of root apical meristem and shoot apical meristem, and it contributes to elongation of the plant, that is lengthening of the plant. Okay, lengthening of the plant is brought about by the apical, lengthening of the root part by root apical meristem and shoot tip by the shoot apical meristem. That is, the shoot apical meristem is present at the tip of the shoot and the root apical meristem is present at the tip of the root. Okay, very frequently asked question, root apical meristem and shoot apical meristem. You can expect. This diagram is given in your NCERT textbook. So please refer it. Okay, refer. You can write over there, refer. Okay, can or cannot be asked. Secondary growth is seen only in case of gymnosperms and dicotyledonous plant because due to the presence of lateral meristem which helps in increase in girth of the organ. So you might have studied in the anatomy chapter about the interfascicular cambium, intrafascicular cambium, core cambium, etc. in the anatomy chapter. All these things comes under the what? Lateral meristems, and that contributes towards a what? Increase in girth of the plant. Is it clear? So next one. So all the concepts are clear in this case, all the concepts. Let's move with the next one. That is, growth is measurable. Growth is measurable. If you take into consideration the cellular level, the growth is measured in increase of the amount of protoplasm. And what is protoplasm? Protoplasm is the physical basis of life. Okay, or the living substance inside the cell is protoplasm. This measurement of the protoplasm is difficult. So increase in protoplasm is difficult to measure directly. So we use certain parameters such as increase in fresh weight, dry weight, length, area, volume, and cell number. Suppose you take into consideration this maize plant, the length or the growth of this particular maize plant is measured by terms of the number of new cells. Is measured in terms of the number of cells. Measured by the number of cells that is formed from time to time. That is, a typical maize root can produce 17,500 new cells per hour in 1 hour. Okay, in 1 hour, the maize plant can produce what? In 1 hour, the maize plant can produce what? In 1 hour, it can produce 17,500 new cells. If you take into consideration the pollen tube, okay, this is the pollen grain and the lengthening of the pollen tube. So the pollen tube growth is measured in terms of its length. That is length. Growth of a pollen tube is measured in terms of length. If you take into consideration about the case of a watermelon, in the watermelon you have measured by the volume. Watermelon is measured with the help of the volume. And if you take into account the leaf based on the surface area, increase in the surface area of the leaf. First it was like this, exchange to be like this, and finally it forms like this, right? So surface area growth of a dorsiventral leaf. So let's move with the next one. Okay, so that was our first topic. Okay, that was our first topic up to the measurement of growth. From this slide onwards, we'll be discussing in our next lecture. So I'll just show you which all slides are left. All these beautiful green slides are there for you to show. So we will be discussing about photoperiodism. Then we'll be discussing about photoperiodism, then vernalization, cold water treatment. So thank you all for your patient listening. So please don't forget to like, share, and subscribe our wonderful channel. So let's meet with the rest of the chapter in our next session. Thank you.